everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by Certara. You may submit questions at any time by submitting them into the questions box of the GoToWebinar platform. This webinar is being recorded and will be available on the AAPS website. It is my pleasure to introduce today's moderator, Suzanne Minton, Director of Content Strategy with Certara. Suzanne, the floor is yours.
Excellent. Thank you so much, Stacy, and thank you so much to AAPS for hosting this eChalk Talk today. Today's presentation is Pharmacokinetic Modeling and Scientific Communications Expertise Advances Sarcoidosis Drug Program. Next slide, please. Got a couple things that we'd like our audience to learn today. We'll be talking about rare disease drug development and why it's so challenging. Our experts will also be discussing population pharmacokinetic modeling and how you can combine it with translational data to help optimize drug dosing regimens for investigational drug candidates. And lastly, we're going to share with you why it's helpful to work with scientific communications experts to help researchers publish their findings more quickly. Next slide, please. We have two very esteemed speakers joining us today. Our first speaker is Nupur Singh. She is currently a Vice President of Marketing and Patient Affairs at Zentria, which is a clinical stage biotech company that's headquartered in Chicago, Illinois. At Zentria, Nupur nurtures their key relationships with patient experts, key opinion leaders, and clinical investigators while leading business development and partnering strategy. Nupur holds a bachelor's degree in kinesiology from the University of Michigan. Joining Nupur is my colleague, Elliot Offman. Elliot is currently a Vice President of Clinical Pharmacology and Translational Medicine at Certara. He has more than 20 years of drug development experience, and his expertise is focused on leading study design, data analysis, and interpretation for a variety of clinical pharmacology studies. Nupur and Elliot, welcome to today's event. I'll now turn it over to Nupur to begin the presentation.
Hi all. So today we plan to discuss a brief background on the XTMAP16 development and place in therapy, an overview of unique non-clinical model for evaluating the efficacy of drugs in treating granulomas and sarcoidosis. And we hope to describe the translational framework for supporting the transition of XTMAP16 from non-clinical into a phase 2. Next slide. I'd like to thank everyone for taking the time to join us today. So Zentria owns XTMAP16, a novel chimeric antibody that targets tumor necrosis factor alpha, or TNF-alpha, a substance in the body that causes inflammation and plays a role in the rare disease of sarcoidosis. In partnership with Certara and researchers at The Ohio State University, we use biosimulation modeling to help estimate the dose and frequency of XTMAP16 in advance of our first clinical trial in patients. Sarcoidosis is a chronic multi-system inflammatory disorder of unknown etiology that is characterized by the presence of non-caseating epithelioid granulomas, accompanied by infiltration of mononuclear cells and destruction of microarchitecture. What we know is that the macrophage-derived TNF-alpha participates in the induction as well as the maintenance of granulomas, and high levels of TNF-alpha released by the macrophages seem to correlate with disease progression. XTMAP16 is a new drug entity with structural differences compared to other anti-TNF antibodies on the market. One difference includes the use of CHO cell line expression system, which may mitigate safety and immunogenicity concerns with this type of antibody. Next slide. In this rare disease landscape, there is a demonstrated need for targeted treatment options for people living with sarcoidosis. Sarcoidosis granulomas are distinct from other granuloma types, and this has implications for treatment. However, drug development in this landscape is difficult. The situation, with a highly diverse clinical presentation and no approved disease-modifying treatments available, there's an urgent need for medications with fewer side effects than steroids, which, by the way, is the current first-line agent and the only FDA-approved medication for pulmonary sarcoidosis. With that, the lack of validated endpoints, the lack of diagnostic markers, and limited real-world evidence, Zentria was forced to explore alternative translational pathways to keep our clinical program built on data and driven by an evidence-based hypothesis. We found solutions through early discussions with KOLs, HCPs, and advocacy groups. This led to us exploring translational efforts for XTMAP16. Next slide. To explore the effect different XTMAP16 doses might have on granuloma growth, we worked with The Ohio State University and Dr. Elliot Krauser's human granuloma model using peripheral blood mononuclear cells, or PBMCs, from patients with active pulmonary sarcoidosis. Briefly, PBMCs from patients with active pulmonary sarc with lung and/or lymph involvement who were naive to tuberculosis were used. PBMCs were then exposed to either uncoated beads or Mycobacterium tuberculosis antigen PPD-coated beads, or purified protein derivative beads, for 7 days. Sarcoidosis PBMCs were then cultured in the presence or absence of inhibitor pretreatments, XTMAP16 or prednisone. The green bars on the right of the graph represent granuloma formation following pretreatment with XTMAP16 at 1, 5, 10, 20, and 40 micrograms per milliliter. The blue bars in the middle of the graph represent prednisone at 2 different doses as well. Both XTMAP16 and prednisone was compared to that following treatment of PPD beads alone. PPD-coated beads, which are the bars in which is the bar in the red, and the uncoated bead, which is the bar in the yellowish green on the left, served as comparable negative controls. The data show that XTMAP16 pretreatment significantly attenuates granuloma formation in the in vitro model in a dose-dependent fashion within the range of 1 to 40 micrograms per microliter. XTMAP16's granuloma suppressing activity at the higher doses was comparable to prednisone, the established first-line therapy for this condition. This translational work marks the first time an in vitro model has informed dose selection and informed trial design for a clinical trial in this rare disease. Utilization of this well-established in vitro model of sarcoidosis enables continued testing of model assumptions as clinical data continues to be available. I will now pass it on to my colleague, Elliot Hoffman.
Thank you, Nooper. So there are a number of different approaches one might undertake to inform human safe and efficacious dose predictions as we transition from non-clinical into human development. This slide illustrates some of the data points and processes one might consider in a translational pharmacokinetic pharmacodynamic, or PKPD framework. In red font, you can see some of the methods that have been applied to the current XTMAP16 development program so far. While today's talk will focus on the potential human efficacious dose predictions in patients with sarcoidosis, translational analysis actually began in the non-clinical phase of development to support the first-in-human dose prediction. In the pre-first-in-human stage, we first evaluated pharmacokinetic exposures which were deemed adequately safe and well tolerated in the animal species, including mouse pharmacology models of disease. We then applied principles of allometry to scale doses to human. Once the PK, or pharmacokinetics in humans, were characterized as part of the first-in-human study, we then leveraged the non-clinical granuloma model described by Nooper and projected doses predicted to be efficacious. The framework we applied will be described in the next few slides. Next slide, please. The basis for the selected PKPD approach, which we have illustrated on the left side of the screen, assumes the effect at the site of action is proportional to the concentration at the site of action, referred here to as the biophase. As illustrated on the right side of the screen, effect entails both safety and efficacy. And to be clear, this was an a priori assumption we made based on the molecular class of the drug and the known pharmacology of XTMAP16. Whenever possible, the translational framework selected should consider what is known about the molecule and the pharmacology to best leverage all available data. Next slide, please. With XTMAP16, pharmacologically, this drug is intended to block a receptor with the intended action of inhibiting an inflammatory cascade of events leading to granuloma formation. Our hypothesis was that if you can identify the concentration which results in a detectable and potentially clinically relevant inhibition of granuloma formation, then the concentration becomes the target concentration at the biophase, which in the case of pulmonary sarcoidosis is the lung tissue. Although I note that granulomas may form in other organ tissues, we're going to focus on the lung today. We leverage the ex vivo granuloma model to estimate a concentration index as the target benchmark concentration in the lung interstitial space. In this case, the IC50, or concentration resulting in 50% reduction in response, which is the granuloma formation measurement, which you see in the top left panel, was used as the benchmark. When developing a novel antagonist or blocker, it is reasonable to infer that the maximal inhibitory effect would result from a drug concentration at the site of action, which is above the inhibitory concentration for the entire dosing interval of that drug. Noting that the IC50 might only inhibit 50% of the effect, selecting a benchmark like the IC90 or some multiple above the IC90 might be warranted. However, it's also important to consider the safety and tolerability thresholds when you're developing a drug. In the case of XTMAB16, we had to balance the need to maximize potential efficacy with the systemic exposure thresholds deemed to be adequately safe. Typically, this means limiting human exposure to not exceed the exposure associated with the no observed adverse effect limit, or NOEL, in a relevant non-clinical species. Next slide, please. This slide illustrates a simplification of a framework applied to predict the human concentration at the intended site of action. Here we leverage Certara's Phoenix nonlinear mixed effect modeling software to fit the first-in-human PK profiles to a compartmental model. Consistent with many monoclonal antibodies, which are administered via the intravenous route, the pharmacokinetics of XTMAB16 followed a typical two-compartment model with linear clearance. One challenge that had to be addressed in this framework was the appearance of AntiDrug Antibodies, which appeared to impact the clearance of XTMAB16 in our healthy subjects. Another challenge presented was, how does one estimate the concentration of XTMAB at the site of action, which is the lung in this case, in order to link the concentration to the effects observed in the non-clinical model? Well, we know the transfer of monoclonal antibodies in the systemic circulation to an organ tissue interstitial fluid occurs via convective transport and transcytosis mechanisms. Sean Betts in 2013 published the estimated antibody biodistribution coefficients for various organ components. And we leveraged this research to predict the interstitial, well, we leveraged the research to utilize the predicted interstitial lung concentration based on the antibody biodistribution coefficient of 15%. And this enabled us to do simulations of XTMAB16 in order to estimate the concentrations that, or the dose and regimen that would achieve adequate concentrations in the lung to inhibit granuloma formation. Next slide, please. Following the modeling, we then produced simulations using a Monte Carlo approach. As described on the previous slide, the serum XTMAB16 concentration was multiplied by the antibody biodistribution coefficient, and the resulting predicted concentration for different regimens were then compared to the IC50, which we obtained by the fitting of the granuloma model. The selected clinical dosing regimens were those that suggested sufficient exposure above the IC50, but yet below the no observed effect limit exposure in a non-clinical species. And this was done with the intention to limit the risk of toxicity in patients with sarcoidosis. Here you can see that the 4 milligram per kilogram every 2-week regimen resulted in trough concentrations above the IC50 for the majority of the dosing interval. While not stated on the plot, the Cmax and AUC predicted here also fell below the exposure at the no observed effect limit. On the opposite end of the scale, the dose intensity of 2 milligram per kilogram every 4-week regimen resulted in concentrations above the IC50 only for a very short duration of the dosing interval, and would only be expected to be minimally efficacious. And now back to Nooper for the next slide.
Thanks, Elliot. In summary, using in vitro data from our phase 1 in healthy volunteers, TNF literature, and allometric and human pharmacology data, The doses and frequencies selected to be explored in our phase 2 study are tailored to sarcoidosis patients. Factoring in the antibody distribution coefficient, the targeted selected doses correlate to those deemed effective in the in vitro granuloma model. The doses are also reflective of a reduction in key inflammatory biomarkers within the model. This is a differentiator for XTMAP16 over other TNF products or current sarcoidosis therapies currently in development. As we develop the protocol to explore XTMAP16's ability to reduce an inflammatory response believed to occur in patients living with sarcoidosis, we also leaned on Certara to help inform the optimal PK sampling time points for our ongoing trial. Noting this rare condition already has a high disease burden, Certara's modeling helped us reduce sampling burden by adjusting to an appropriate PK sampling schedule that still provides Zentria as the sponsor enough data to adequately characterize the PK at this phase of study. We've learned to value translational work in rare diseases where real-world evidence lacks. As a sponsor, collaborations like ours with the Ohio State University and Certara are imperative in bridging real-world evidence gaps and moving the disease state science forward. Modeling will continue to be leveraged to explore potential racial and ethnic PK differences and to help manage risk and accelerate the potential of this investigational product throughout the life cycle of clinical development. Elliot? Next slide.
Next slide. Yeah, thank you. In summary, XTMAB is a monoclonal antibody, and it's an inhibitor of TNF-alpha engineered to reduce activation of an inflammatory pathway implicated in the pathophysiology of sarcoidosis. Utilization of a translational non-clinical model of granuloma enabled the estimation of potentially efficacious XTMAP16 concentrations at the site of action. Through PKPD modeling, we have leveraged the non-clinical models of efficacy and safety along with early human clinical data to guide the development of a phase 2 protocol for the treatment of patients with sarcoidosis. And the work here today, described today, can be found in our publication in Frontiers in Pharmacology, and that's noted on the right side of the screen. I'd also like to acknowledge my Certara colleague, Amy Porter, of the Certara Medical Communications team for supporting the writing of this manuscript, which has been critical in communicating the rationale and justification for the dose regimen selected for the phase 2 protocol to the physician community. In general, publications of this nature lend to transparency of data informing clinical protocols and enable the physician community to learn about the data in a concise and well-communicated manner without having to dive into the esoteric modeling and simulation reports. We hope you have found this session informative, and we welcome any questions. Thank you for your attention.
Fantastic. Thank you so much, Nooper and Elliot. As Elliot mentioned, we invite our audience to submit their questions to our speakers in the questions panel, and it looks like we already have our first question. I'll direct this to you, Elliot. So it says, the response to the monoclonal antibody appears to be very steep. Is this a concern from a safety point of view?
Well, it can be as part of any translational and human analysis. We have to do the assessment of safety in humans. I will note that we didn't see any acute safety and tolerability concerns after single doses. But if you truly want to go start low and go slow in patient development, and to that end, and to address some of that uncertainty, there is a sort of a dose finding component to the phase 2 study, rather than barreling right into a large group of patients with sarcoidosis. There's a lead-in dose finding which will enable us to make estimations about the safety and tolerability. So I'm not sure if that fully addresses the question, but we tried to build in the phase 2 program a means of assessing that, and maybe Nooper, you could speak a little bit more to that particular aspect.
Sure. We're aware that the in vitro work was completed with a high concentration of purified PBMCs. And so the level of those PBMCs, but the lung concentration is unknown for the patient. And so we don't know exactly how much influence we'll actually have in the lung. But using the modeling data, we're using the data that's available before we get into patients, and then we'll start to adjust the model as data comes in.
Makes sense. Looper, could you talk a little bit about some of your thoughts on the future directions for this program?
Sure. For us at Zentria, continued conversation, close partnerships with all of our existing partners, but also the key opinion leaders and researchers. We hope to continue to foster those and build upon those. As a data-driven approach is needed for any rare disease clinical development model, we also hope to lend the data of this large randomized clinical trial to furthering the research of sarcoidosis, so it can help all community members. We will pivot from, of course, our phase 2 and our phase 3, but we're exploring a lot of different biomarkers and endpoints in this phase 2 to hopefully build a stronger phase 3 that, of course, is data-driven. But we're very honored and excited to be in this community of strong researchers and advocates. So we hope to continue those collaborations moving forward.
Great. Elliot, XTMAB16 is a biologic drug, so one would expect the formation of anti-drug antibodies. How did you account for anti-drug antibodies' impact on XTMAB16's pharmacokinetics when you performed simulations?
That's a great question. So while we were evaluating our data, it became very clear that we had some unusual looking pharmacokinetics. When you see that and you don't have your anti-drug antibody data available to you, it's difficult to infer that, and you might partition that answer towards a nonlinear clearance mechanism. So here we were fortunate enough that we both had results that demonstrated that there was anti-drug antibody formation, and in fact they were also neutralizing antibodies. So we have two approaches that we can use. We can assess both at a sample level and at a patient or subject level whether or not anti-drug antibodies are affecting the clearance or other PK characteristics of the drug, and we tested both. We found in this particular case the subject level positive ADA characterization was sufficient to describe that, and then so subjects were then either noted to be ADA positive or ADA negative, and so we then have a clearance adjustment factor we can apply for subjects who are ADA positive, and they're going to have theoretically lower systemic exposure. So then when we performed our simulations, we performed simulations both for subjects who are ADA positive and ADA negative. And from a safety perspective, you want to look at the ADA negative because that sort of represents the high end of the exposure. And for potential efficacy purposes, we want to take into account those who might be ADA positive because they might be expected to have lower systemic exposure. And we want to make sure that we've accounted for subjects or patients that might have lower systemic exposure when trying to come up with that dosing regimen.
Awesome. Thank you, Elliot.
Yeah, thank you.
Got a couple more questions that came in. Nooper, I think this might be one for you. Someone says they wanted to know if any histology studies were done on the effects of biodistribution of XTMAB16 on organs and tissue in preclinical species.
They were. We have not published on that, but we did do tissue cross-reactivity in our non-clinical work.
Okay, fantastic. Thank you, Nooper. I think this may be a question for Elliot. Do you have a preclinical disease model to determine a reasonable efficacy threshold? You mentioned that the IC50 determined in vitro might not be sufficient.
Well, I mean, as Nooper mentioned, there have been pharmacology models of disease that were assessed, but that data has not yet been published. So we have to be careful about what we can articulate. But suffice to say that the exposure associated with efficacy in the mouse disease models is in line with what we might have expected from the in vitro models. So they do align in any translational analysis. And I think this is just sort of this general statement. It's always nice to have different lines of inquiry and investigation sort of intersect at some point to support your hypothesis. And in this particular case, we had more than one line of evidence that that suggested in and around the same effective concentration level.
Great, thank you. I think we've got time maybe for one more question. Maybe you can take this, Nooper. So sarcoidosis, you talked at the beginning about how it's a disease that affects different organs, and in this case study we looked at the lungs. Can you talk about, is Zetria doing any work to look at granuloma formation in other organs?
We are. So our clinical trial in patients allows for some extrapulmonary manifestations. Primarily patients have sarcoidosis in their lungs, but there's then also a high likelihood that there's granuloformation formation on other organs. Following the lungs, the lymphatic system, the skin, and the eyes are often implicated. So we are including some of those subsectors, but I'm sure as everyone here knows, we need to have as much of a consistent patient profile as we study. So we'll be allowing some extrapulmonary, but excluding cardiac sarcoidosis primarily from this study. But we hope to continue exploring and diving into some of the other indications to get more data around that, especially because depending on the level of diagnosis and the level of sort of knowns that a patient has, patients present with a wide spectrum of both systemic and non-specific symptoms that might not be linked to any specific organ. So we're trying to collect as much data as possible, but enough that we can sort of make those correlations come time of analysis.
Fantastic, that makes so much sense. So I'd like to thank our speakers today, Nooper Singh and Elliot Offman, for this really excellent and educational presentation. I'd also like to thank AAPS for hosting this event and our audience for their attention and these excellent questions. I'm now gonna turn it back over to Stacy to wrap up the webinar.
Thank you so much for a wonderful presentation, and thank you to Certara for always partnering with us and for today's webinar. And we look forward to seeing you all again at a future eChalk Talk. You may now disconnect from the webinar. Have a wonderful day.